The human body consumes roughly 250 milliliters of oxygen per minute at rest. That number sounds modest. Yet imagine every cell burning fuel continuously like a light bulb that must never go dark. The heart pumps blood. The lungs exchange gases. And the brain, though only about 2% of body weight, uses nearly 20% of the body's total energy. Everything runs smoothly, like a brightly lit city at night. Then a molecule appears, made of just carbon and nitrogen. Colorless. Effective in small amounts. It does not destroy the lungs. It does not instantly stop the heart. Instead, it does something more precise. It locks the central power station. Cyanide does not kill by removing oxygen from the air. It prevents cells from using the oxygen that is already there. You continue breathing. Your blood remains red, but the power plants inside your cells shut down. That is what makes this compound a classic example in physiology and toxicology. To understand what happens, we move inside the cell where the real story unfolds. Each human cell contains hundreds to thousands of mitochondria. Under an electron microscope, a liver cell reveals oval-shaped structures with folded inner membranes. These folds increase surface area for the electron transport chain, the system responsible for producing ATP, the body's energy currency. Under normal conditions, oxygen serves as the final electron acceptor in this chain. Electrons pass through a series of membrane proteins and eventually reach an enzyme called cytochrome C oxidase. When oxygen accepts electrons and protons, water is formed. This process generates an electrical gradient across the mitochondrial membrane, similar to charging a microscopic battery. ATP synthase then uses that gradient to produce ATP. Cyanide interferes at the final step. The cyanide ion binds to the iron within the active site of cytochrome C oxidase. Once that site is occupied, oxygen can no longer bind. The electron transport chain halts. The electrical gradient collapses. ATP production stops. The critical detail is that oxygen is still present in the bloodstream. Measured oxygen levels may not drop immediately, yet cells enter a state of internal suffocation. If a brain cell lacks sufficient ATP for just a few minutes, irreversible dysfunction begins. The brain is especially vulnerable because it consumes energy continuously and has almost no reserves. No mechanical force is required. No tissue must be torn. It is a molecular lock compact, specific, and fast. What happens at the organ level when this energy supply is cut? As ATP levels fall, ion pumps embedded in cell membranes can no longer function effectively. Sodium and calcium accumulate inside cells, disrupting membrane potentials. In heart muscle, altered electrical balance can trigger arrhythmias. In the brain, neurons lose their ability to transmit signals properly. Time becomes the decisive factor. The brain can tolerate oxygen deprivation for about 3 to 5 minutes before irreversible injury begins. The 5-minute mark is often emphasized in emergency training because beyond it, the risk of permanent brain damage rises sharply. For the heart, energy interruption quickly weakens contraction. In cyanide poisoning, one distinctive feature is that the skin may remain pink or even appear unusually flushed. Oxygen is not being fully used by tissues, so venous blood can remain relatively oxygen-rich. This differs from many other forms of suffocation, where oxygen is actually absent from the air. The body responds by increasing breathing and heart rate in an attempt to compensate. Yet when the target enzyme remains blocked, increasing oxygen delivery does not solve the underlying problem. The city has more air, but the power plants are still offline. In industrial contexts, cyanide has been used in gold mining because of its ability to form complexes with metals. In biology, that same capacity to bind metal ions underlies its toxicity. The same chemical property, two entirely different outcomes. Given the rapid progression, a reasonable question follows. Can the circuit breaker be switched back on in time? Treatment for cyanide poisoning focuses on two strategies: removing cyanide from its enzyme target or converting it into a less toxic form. One commonly used therapy is hydroxycobalamin, a form of vitamin B2 presache. This molecule contains a cobalt ion at its center, which can bind cyanide to form cyanocobalamin, a compound that can be safely excreted by the kidneys. Hydroxycobalamin acts as a mobile molecular trap. Instead of allowing cyanide to attach to cytochrome guanine C oxidase, the cobalt within the medication attracts and binds it. This is not magic. It is chemistry based on the affinity between metal ions and cyanide groups. Historically, another approach involved the use of nitrites to induce the formation of methemoglobin. Methemoglobin contains iron in a different oxidation state and can bind cyanide, drawing it away from mitochondrial enzymes. However, this method requires careful management because methemoglobin does not transport oxygen as efficiently as normal hemoglobin. Speed of intervention directly affects outcome. In industrial settings or structural fires where cyanide may be produced from burning nitrogen-containing plastics, emergency training emphasizes rapid recognition and treatment. What makes cyanide such a powerful teaching example is not only its danger, but how clearly it demonstrates the body's absolute dependence on cellular respiration. Block a single enzyme, and the entire energy system collapses in sequence. Cyanide does not rely on force. It only needs to occupy the right molecular position within a critical biological reaction. This discussion is not meant to provoke fear, but to highlight the precision of molecular biology and the importance of every link in the energy chain. Once the mechanism is understood, familiar words like breathing, oxygen, and energy take on new depth. If these invisible circuit breakers inside the body spark your curiosity, the next exploration might open another door in toxicology, where dosage, context, and biological design determine every outcome.